this video, I am going to explain the urea cycle. What is urea cycle? It is the first metabolic cycle. It is the first metabolic cycle and it is elucidated by Hans Krebs and Kurt Hensleit. It is the first metabolic cycle and discovered by Hans Krebs and Kurt Hensleit. So it is also known as Krebs-Hensleit cycle. Krebs-Hensleit cycle. So urea cycle is the first metabolic cycle. It is discovered by Hans Krebs and Kurt Hensleit. So it is also known as Krebs-Hensleit cycle. During the amino acid metabolism or protein metabolism, urea is formed as the end product. So it is the end product of protein metabolism. Urea is the end product of protein metabolism. So in our body, the nitrogen of the amino acid is converted to ammonia. In our body, the nitrogen of amino acid is converted to ammonia. This ammonia is toxic to our body. So it is converted to urea. This urea is the non-toxic. So the nitrogen of amino acid is converted to ammonia, that is toxic to our body. So it is converted to urea and detoxified. It is converted to urea and detoxified. Urea is synthesized in the liver. Urea is synthesized in the liver and it is transported to the kidney for the excretion. So urea is synthesized in the liver and it is transported to the kidney for the excretion. In the nitrogen containing compounds excreted through urine, about 80 to 90% is urea. In the nitrogen containing compounds excreted through urine, about 80 to 90% is urea. Now coming to the structure of urea. This is the structure of urea. In this we can see two amino groups and one carboxyl group. So this urea contains two amino groups and one carboxyl group. So in this amino group coming from ammonia, one amino group coming from ammonia and the other is from the aspartate. It contains two amino group. One is from the ammonia and other is from the aspartate. And the carbon atom from the carbon dioxide. So urea contains two amino groups. One is from the ammonia and one from the aspartate. And the carboxyl group or carbon atom from the carbon dioxide. Now coming to the cycle. This is urea cycle. This is a 5-step containing cycle. 5 steps are there in the urea cycle. In the urea cycle, 5 steps are there and 2 steps takes place in the mitochondria and the remaining 3 takes place in the cytosol. So it contains 5 steps. This cycle contains 5 steps. 2 of them in the mitochondria. And the remaining 3 is in the cytosol. Now coming to the first step of the urea cycle. First step is synthesis of carbamyl phosphate. Synthesis of carbamyl phosphate is the first step in the urea cycle. This is the first step in the urea cycle. Here the carbamyl phosphate synthase 1 enzyme which is present in the mitochondria. The carbamyl phosphate synthase 1 which is present in the mitochondria catalyzes the condensation of carbon dioxide and ammonia. To form carbamyl phosphate. Once more, the carbamyl phosphate synthase 1 which is present in the mitochondria catalyzes the condensation of carbon dioxide and ammonia to form carbamyl phosphate. This reaction requires 2 ATP molecules, and 2 ATP molecules are converted to 2 ADP and inorganic phosphate. This step is an irreversible step. This is an irreversible step, and this enzyme, the carbamyl phosphate synthase 1, require NAG, that is N-acetyl glutamate, for its activity. So, the carbamyl phosphate synthase 1 requires N-acetyl glutamate for its activity. Then, it is an irreversible reaction. So, this is the rate limiting step in this cycle. This is the rate limiting step in this cycle. Now, coming to the second step. Second step of this cycle is formation of citrulline. Second step is formation of citrulline. In this step, the ornithine combines with the carbamyl phosphate. Ornithine combines with the carbamyl phosphate in the presence of the enzyme ornithine transcarbamylase. Ornithine transcarbamylase to form citrulline. Citrulline and ornithine are basic amino acids and are not present in the structure of protein due to lack of codons. And the ornithine is regenerated in the cycle, and the citrulline synthesized is transported to the cytosol. The citrulline synthesized here in the mitochondria is transported to the cytosol. The transport of the citrulline is through a transporter system. Transporter system. The third step of the cycle is synthesis of arginosuccinate. Synthesis of arginosuccinate. In this step, the citrulline condenses with aspartate. The citrulline condenses with aspartate in the presence of the enzyme arginosuccinate synthase. The citrulline condenses with aspartate in the presence of arginosuccinate synthase to form arginosuccinate. So, in the third step, the citrulline condenses with aspartate in the presence of arginosuccinate synthase enzyme and forming arginosuccinate. Here, 1 ATP is required, and that is converted to AMP and PPI, inorganic pyrophosphate. The second amino group of urea is incorporated in this step. The second amino group of urea is incorporated in this step, that is from the aspartate. The fourth step of the reaction is cleavage of arginosuccinate. Cleavage of arginosuccinate is the fourth step of the urea cycle. In this step, the arginosuccinate is cleaved by the arginosuccinase enzyme. The arginosuccinate is cleaved by the enzyme arginosuccinate to form fumarate and arginine. Arginosuccinate is cleaved to fumarate and arginine by the enzyme arginosuccinase. So, this arginine acts as the intermediate for the urea cycle. At the same time, this fumarate liberated is act as the connecting link with the TCA cycle. The fumarate liberated act as the connecting link with the TCA cycle. At the same time, the arginine act as the intermediate for the synthesis of the urea. The fifth and final step of the cycle is formation of urea. So this is the last step in the urea cycle. Here the arginine is cleaved by the enzyme arginase to form urea, and ornithine is regenerated. Here the arginine cleaved by the enzyme arginase to form urea and ornithine. That is, ornithine is regenerated. This regenerated ornithine is again utilized for the next urea cycle. Similarly, the urea is the end product in this cycle. So this is known as the urea cycle. Here the arginase enzyme, which is mostly found in the liver. Arginase enzyme mostly found in the liver. But all other enzymes of this urea cycle are present in other tissues also. But arginase present only in the liver. So the urea cycle takes place in the liver. So the urea cycle takes place in the liver. This arginase enzyme is activated by cobalt and manganese. The arginase enzyme is activated by cobalt and manganese. This is the overall reaction of the urea cycle, that is ammonia plus carbon dioxide plus aspartate plus 3 ATP gives urea plus fumarate plus 2 ADP 2 PI plus AMP and PPI. This is the overall reaction of the urea cycle. This cycle is irreversible. The urea cycle is irreversible and it consumes 4 ATP. It consumes 4 ATP. 2 is for the synthesis of carbamyl phosphate. Carbamyl phosphate. 2 is for the synthesis of carbamyl phosphate. And the other one is utilized to produce arginosuccinate. At that time, ATP is converted to AMP and PPI. So it is actually equal to 2 ATP. So this urea cycle consumes 4 ATP. Once more, 4 ATP is utilized for the urea cycle, 2 ATP for the synthesis of carbamyl phosphate and 1 ATP for the conversion of arginosuccinate. Production of arginosuccinate, which is equal to 2 ATP because the ATP is converted to AMP plus PPI. So it is converted to 2 ATP. So actually 4 ATP molecules are consumed for this cycle. This is about the urea cycle. Thank you.